Hello, college biology. Today, we are going to start our series of videos on cellular respiration. And so I'm going to show you something that you may have seen before from your general biology. This is the generic reaction for cellular respiration. We have our C6H12O6, our glucose. We have 6 molecules of oxygen. We produce 6 molecules of carbon dioxide, 6 molecules of water, and ATP. Well, la-di-da, isn't that happy times? That energy is in ATP. How good is this? This is a lie. If you remember your balancing equations from general chemistry, then you'll know that ATP, if it's on this side, it's got to be on this side. And we can count up our CH's and O's, and we do a fine job of CH's and O's, but we do not have the parts for ATP over here. So let's fix that. Okay, let's do that. We have glucose, we have 6 oxygen, we have 32 ADP, we have 32 phosphates, PO4, phosphorus surrounded by 4 oxygen, and then we yield 6 carbon dioxide, 6 water, and 32 ATP. Well now, how do we feel about that? That's much better. It's balanced. It's good. It's even on both sides. This has got to be the real reaction for cellular respiration, right? No. This is also a lie. I'm going to show you the dark truth of cellular respiration. I'm going to show you that it is far more complicated, because cellular respiration really looks like this. You start with glucose, and it undergoes a reaction to become glucose 6-phosphate, which then undergoes a reaction to become fructose 6-phosphate, which this undergoes a reaction to become 2 glyceraldehyde 3-phosphates, which each undergo a reaction to give us a total of 2,1,3-biphosphoglycerates, which then undergo a reaction to become 3-phosphoglycerate, which then undergoes a reaction to become phosphophenol pyruvate, which then undergoes a reaction to become pyruvate, which is phase 1. Then there's a second phase where this pyruvate is further altered as it enters the mitochondria into acetyl-CoA. And then phase 3, the acetyl-CoA enters the Krebs cycle, sometimes called the citric acid cycle, and it becomes citrate, which then becomes ketoglutarate, which then becomes succinate, which then becomes fumarate, which then becomes oxaloacetate. I mean, I can hardly pronounce all this stuff. And then it just keeps going in this cycle, so you'd think, oh my gosh, we're trapped in here forever. Except during this process, we're making NADH, NADH, and FADH2, which are then used here in phase 4, the electron transport chain, which then the NADH gives away their electrons, which pumps hydrogen out into the environment of the intermitochondrial space, which is then used as the proton motive force to fuel ATP synthesis to make ATP. And that is all of the reactions of cellular respiration. So, let's go back to this. This simple reaction is a lie. It is a gathering of all that you need and all that you get out of cellular respiration from all of those steps in the chemical reactions. We're going to look at the big picture right now. Big picture. Glucose is oxidized to charge electron carriers. What the heck does that mean? Oxidation is a fancy word that means something is acting like oxygen. Well, oxygen is highly electronegative and it pulls away electrons. So if glucose is oxidized, the electrons have been ripped away from glucose. When we break glucose down, the reason why our cells do that is really to get the electrons out of it. So we're stealing electrons away from glucose, which they're negatively charged. They have this electromagnetic energy that they can use since opposites repel, things of that nature. So we're going to take those electrons, we're going to use them later, and we need to store them in electron carriers. So we break down glucose to put electrons with electron carriers. Then, what do we do with the electron carriers? The electron carriers are used to force transport proteins to pump hydrogens. These are transport proteins that need the electromagnetic polarity of a negative charge of that electron to change their shape to isomerize, and when they do that, they push a proton through the membrane. So we need to break down glucose so that we can push hydrogens across a membrane. That's what we want to do. We want to get the hydrogens out of here. So let's put them across the membrane using the energy from glucose, which we got from its electrons. And finally, why do we need to move those hydrogens out there? The hydrogens are used to interact with a protein, an enzyme called ATP synthase that actually makes ATP. So, in summary, glucose, we ripple your electrons away. We use your electrons to move hydrogens. We use the moved hydrogens to make ATP. That's the big picture. Get electrons from glucose, move hydrogens, use hydrogens to make ATP. That is all for this video. We will do much more in the future. I hope this helps. Struggle.